Okay, so we'll start chapter 6 on chromosomes and cell division with some kind of basic background on chromosomes and how they're used in the cell and different types of cell division. We've already talked about in chapter 3 the two basic types of cells. We talked about prokaryotic cells and also eukaryotic cells. So prokaryotic cells would include things like bacteria, and their method of division is very simple. They use something called binary fission, and we'll look at some diagrams of that in a minute. But in binary fission, bacteria can accomplish that very, very quickly. As an example, since we're all taking class in a micro room this year, it's nice to know how quickly bacteria can divide. Something like E. coli in the laboratory setting with no other bacteria that's competing for the nutrients, it can divide once every 20 minutes. So it only takes 20 minutes for every single cell to divide. So if you start with one cell, in 20 minutes you'd have 2, and then 4, and then 8, and they would double like that every 20 minutes, which makes it really nice in a micro lab because you can get millions of cells in a couple of days, makes it easier to study. Eukaryotic cells, they still have to divide, but they do it slightly differently and depending on what the cell is trying to accomplish. So cells can go through either mitosis or meiosis, and mitosis is used just for growth and repair, things like that. Meiosis is a special type of cell division only used for producing gametes during reproduction. So looking again at comparing prokaryotic and eukaryotic cells, focusing in on the chromosome, and we've looked at both types of chromosomes again in chapter 3, and we know that prokaryotic cells like bacteria, they just have one big loop of DNA, and that DNA can be quite substantial inside the cell. E. coli, again, kind of a generic organism as a model, its DNA normally fills about half the interior of the cell. So it basically just contains DNA inside of it. Even though that's one big loop, it takes up a large proportion of the cell. Eukaryotic cells are very different from prokaryotes because instead of having one big loop of DNA, they have multiple linear chromosomes. As an example, humans have 23 pairs, or a total of 46 individual strings or strands of DNA that are chromosomes. And because they're linear, they have ends hanging out loose at the end, and we'll talk about those in a minute. And also, to compact all of that DNA inside the cell and prevent it from getting tangled or broken, eukaryotic cells also have small proteins that the DNA wraps around. You can see that here at the end of this figure. Those orange dots are the histones, and the DNA will wrap around those as it packs in tightly into the chromosome structure, and that prevents the DNA from getting broken and damaged, because DNA really is a very long and fragile strand. One other thing about eukaryotic cells that's important to remember, and we'll come back to this, because they're linear, I'll just draw a little piece of DNA down here. Because they're linear, those ends are exposed. And to prevent enzymes and other things or damage from occurring to the DNA, there's special DNA sequences at the ends of all the chromosomes that are called telomeres. And those telomeres have special structures which prevent things from chewing them to pieces. However, every time the cell divides, it loses a little piece of that telomere at the end. This is also why telomeres are really important, because as it loses those little pieces and gets into some critical regions closer to the DNA, once those kind of lost pieces of telomere hit that critical region, it triggers cell death. So the cell gets a message, says, hey, we're getting too old, we're going to start damaging important DNA. So it sends a message actually to the lysosomes, which we talked about in chapter 3, and tells the cell to digest itself from the inside out, and it dies off. That's part of what causes aging. So that's why as you get older, your skin starts looking thinner. I don't know if you've ever noticed, in very old people, their skin looks very papery and thin, almost like you could see right through it. It's because it's not as thick. They have fewer cells producing skin. That's also why things start to sag as you get older, because you have fewer cells holding it all together. And so that helps contribute to aging. And each cell line, on average, can only go through about 50 cell divisions. Now, some cells can do many more. So stem type cells in your skin, they're going to divide many more times than 50. Other cells, like your brain cells, they're basically never going to divide at all after you reach childhood. So that's an average number of cell divisions across all the different cell lines of your body that do divide, at least sometimes. A problem can occur with those telomeres, and there are certain types of syndromes where something goes wrong with the telomeres and they don't get reset properly when they're passed on from parent to offspring, and it can lead to things like Hutchinson-Gilford progeria. And this person over here in the picture, if you notice, he's got big glasses, he's kind of got wrinkles, and if you were to talk to him about his medical history, he's on high blood pressure medication, he's got cardiovascular disease, he's got arthritis, all of that. However, that is an 8-year-old child. The reason he's aging incredibly rapidly is found in his chromosomes. So here's his chromosomes. If you look at the very ends of each of those chromosomes, you can see little yellow spots. Those are the telomeres. At 8 years old, those telomeres should be substantially longer than what they are now. So he started out, because of this defect, with only partial telomeres. None of this was there when he was born. So he aged very, very rapidly because, before he was 5 years old, it was starting to get into this region on some of his cells. Another thing, which unfortunately is very common with telomeres, they're also involved with cancer. So cancer cells, if they continued to allow the telomere to function properly, there's our telomere again, if a cell became cancerous, then we would just have to wait for it to run down the clock, and then the cell lines would die off and the cancer would just simply disappear. However, in cancer cells, I think it's something like 98% of cancers, so a huge proportion of cancers, their cells become immortal, and we'll talk about that a little bit more later. So wherever they were at the point where that cell became cancerous, they stop clipping their telomeres, and many types of cancer actually regrow new telomeres, so kind of turn back the clock on the lifespan of the cell. This is what allows cancer to continue to grow and grow and grow and cause biological problems. And we'll talk more about cancer later on in this chapter. So looking at prokaryotic cells again, we want to go into the details of binary fission. Binary fission is an asexual type of reproduction. So you take one bacterial cell, it copies its DNA, divides in half, and then you have two. Technically, those two new cells are daughter cells, and each of those can go on to divide again, but they haven't exchanged any DNA. So it's asexual reproduction. 3 basic steps of binary fission, I've already kind of mentioned them once. You need to copy the DNA. It's just a big loop of DNA, so it starts on one side and begins copying in both directions until it's made it all the way around the circle. Now we have 2 separate circles of DNA. Those 2 circles of DNA will then migrate or move a little bit or get attached to either end of the cell. The cell will stretch just a little bit to give it space to divide in half. So remember, E. coli, normally that DNA takes up half the interior space of their cell, so they need to make a little bit of room in order to make that division between 2 cells. In fact, if you look at this actual photo of a cell up here, you can see all this clumpy orange stuff. That is the DNA. So it's just packed with DNA. Once it's elongated enough to give it space, it divides. So copy, elongate, and divide. Those are the 3 steps of binary fission, and that's how things like bacteria divide. Eukaryotic cells are a bit more complicated. As we saw even in chapter 3, they have more parts to them. So eukaryotic cells go through what's called a cell cycle. They start with interphase, and during interphase, and it's the purple part of this graph over here, during interphase, the cell is just functioning as a cell. So a majority of the time, your cells are in interphase. So they're just acting like skin cells. They're just acting like liver cells. They're just being muscle cells, and they're in interphase. So that's the functional phase and where cells spend most of their time. However, if a cell gets a hormonal signal that it needs to divide, maybe, some skin got sloughed off, so the skin cell needs to divide, there's space in the body, the cells need to divide, then it will continue in the process and move through G1 is where it was during the functional phase. Once it gets a signal to divide, it will leave G1, just functioning as a cell, and copy the DNA. So DNA synthesis. If you notice over here, we have one blue strand, one green strand, and one orange strand, and that's the way the cells normally are. But when they copy the DNA, now we have two blue strands held together, and we have two green strands also anchored together, and two different orange strands held together. So now we've synthesized or copied our DNA. I should mention that some cells in the body never leave this first gap 1 or G1 functional phase, like your brain cells. They've stopped dividing very soon after birth, and they're never going to divide again. So instead of ever continuing, they're kind of stuck in another loop here called G0, where they just continue to function as a cell, and they never divide. Things like your skin cells are constantly dividing. So we move on to our DNA synthesis. Once the DNA has been copied, then the cell needs to rest and recover a little bit, and also just function as a cell for a while until it has enough energy. Hormonal signals are still telling it, hey, we need the cell, you can divide. Then it enters, or it's in gap 2. After gap 2, it will enter mitosis. And mitosis is just the basic type of division which is used by somatic cells. So mitosis is used by somatic cells, and that just helps with growth and repair. So all of your growth from in your mother's womb to now, all of the repair that your body's done, so cuts, scrapes, bruises, all of that just uses mitosis. So mitosis, it takes those two strands of DNA that we've produced, and it separates them. And we'll go through the stages of mitosis. The last part of the cell cycle is cytokinesis, and cytokinesis is division of the cytoplasm. So kinesis is division, cyto refers to the cytoplasm, so this is just cytoplasm division to separate these two individual cells. So that's what somatic cells do in mitosis. Germ cells go through a slightly different process. They go through meiosis, and that only happens in reproduction to make gametes, and gametes would be the sperm if you're a male, or eggs if you're a female. So when I refer to the gametes, it's either sperm or eggs depending on the gender of the organism. So mitosis is only used in somatic cells for growth and repair. Meiosis is only used in germ cells for reproduction. Let's review a couple concepts that are fairly basic on DNA. So how does a cell accomplish DNA replication? Well, we have in this figure the original strand of DNA at the top, and DNA replicates in a semi-conservative fashion, which means it takes the original strand of DNA and half of it serves as a template, so we keep half of the original on both sides, and then we make a new strand to match on the opposite side. It's semi-conservative because it keeps half and makes brand new half every time the cells divide. That's also a very good mechanism to prevent errors or mutations or problems in the DNA because DNA is also complementary. It's made up of 4 different bases: adenine, cytosine, guanine, and thymine, referred to as A's, G's, C's, and T's. And in this diagram, they're color-coded, and you'll notice that yellow always pairs with green. So anytime you see yellow, it's always going to have a green across from it. So our C and G will always pair with each other. So G pairs with C. And same thing for A and T, they're always going to pair. So anytime you see pink in this diagram, you also see blue. And that's true on both of our semi-conservative replications here. So A always pairs with T, and G always pairs with C. You'll need to remember that for the test, and a really easy way to remember that is the shape of G and C are very similar, so they go together. So if you can just remember that, and then the other two get paired as well. I will probably on the exam give you something like this. And ask you to tell me what is the complementary half of that DNA strand. So what you would need to do is go through and look at each letter and tell me what it would pair with. So C would pair with G, correct? G would pair with C. A is going to pair with T, T with A, and so on through the remainder of the strand. So make sure that you're able to link together complementary bases so that you're able to do that on the exam. A little bit more about mitosis, because that's what we're going to go into on the next lecture. Mitosis, again, is only used for things like growth. So from a single cell that was produced when sperm and egg combine, all the way to the trillions of cells that make up your body today, that was all accomplished using just mitosis. Also for replacement, so anytime your cells get damaged, so if you get a scratch on your arm, the cells surrounding that scratch get chemical signal that says, hey, there's no cells here next to us anymore, and so they're actually going to start to grow and slide over and cover that open wound. So replacement. Other places that get replaced, so we know about skin cells sloughing off, right? So you've been watching this video for a few minutes now, and you lose several million cells every minute. So you have piles of millions of cells sitting around you. If you were to go around with a little piece of sticky tape and collect those cells, put it under a microscope, you'd be able to find your skin cells all over the place, just surrounding you. Skin cells actually is what makes up a majority of dust. It's just skin cells that have been shed. That's also how dogs are able to track people, just following the trail of skin cells that they leave behind. And so all of those lost cells need to be constantly replaced. Other places that you would lose skin cells would be your entire digestive tract. So this young man here is eating spaghetti. But if you think of something that's crunchy, like a cracker or a chip, you chew it up, and have you ever swallowed a chip and felt it kind of scratch your throat on the way down? Well, that chip is going to continue to scratch and scrape cells off your digestive tract all the way down your throat into your stomach and all the way through your small and large intestine out the other side. So it's pulling cells off along with it as it goes through. Because of that, your digestive tract needs to constantly be making new cells. So on average, you get an entire new lining for your digestive tract about every 3 days. All of those cells have been replaced. Other things, if you've damaged your cells, this gentleman over here is shaving, so he's damaging his cells, so they're going to start to grow back and replace each other. And also apoptosis, so just programmed cell death, where cells just die off. They may need to be replaced as well. So that's the end of this first part of the lecture. Remember to read the textbook for this first section of the lecture, and let me know if you have any questions.